Hi, this is Abby Chavez. I did this first assignment by myself because I was a little lazy and I didn't get my group in time. So here we go. So the first question, suppose you perform the following cross. Assume that all of these genes undergo independent assortment. So for this first question, I kind of duped up. So I just watched the forked line method video, and then I did all the work, and then I thought that it would suit me to make like the whole chart. As you can see, I kind of gave up toward the end, and then right here I started to do all the math together. So I did like one, and then I wrote 4 others out, and then once I got to here, I kind of realized that I didn't need to do that, and I just needed to go to here, and then for example, for A, it asks what proportion of the offspring is expected to have this genotype, and so I just took this genotype, found all of the ones over here, and then took the percentages, or the fractions, I guess, or the ratios, there we go, and then I found the percentage, and then that's what I did for each of the rest of ones. For letter C, it asks for all of these ones, but then for this one, it didn't have that over here, and so you couldn't do it. So there is no. For number 2, it says to fill in the blanks. So that's what I did. I filled in the blanks. So it says paired homologous chromosomes are often called sister chromatids or tetrads. B says the structure that connects the centromere to the microtubules of the meiotic spindle is called a kinetochore. Pretty sure that's how you say it. C, a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 8 sister chromatids, while a fruit fly cell in metaphase 2 has 16 sister chromatids. D, it says a Punnett square is a type of genetic cross that can determine if a purple flowered pea plant has the dominant or the heterozygous genotype. And then E says a non-functional allele is sometimes called a null allele. While a partially functioning allele is called a silent allele. For this one, I wasn't too sure, but I'm, like 65% sure that's what it is. For F, Mendel's law of independent assortment involves 2 traits, while Mendel's law of segregation involves a single trait. For C, it asks, let me scroll a little up on the computer here. It says, suppose an organism has the genotype of that one. I have to practice reading or being able to say the names. Assume that the alleles are assorted independently. So the first one, A, it says diagram all possible homologous chromosome arrangements in this organism during metaphase 1. Be sure to indicate that alleles are found on each pair of homologous chromosomes. So for 3, this one was kind of the one that I was having the hard time with. And since I was doing it by myself, I didn't really have too many people to ask. And so I actually went on and I watched other people's videos, so hopefully I don't get in trouble for that. So for A, it asks what I just said. So basically for the first one, you have the T's stay the same, both of the R's stay the same, and then the Y's are the same. For the second one, the T's are the same, the R's are switched, and the Y's are the same. For the third one, the T's are the same, the R's are the same, but the Y's are switched. And then for the last one, the T's are the same, the R's are switched, and then the Y's are switched. So this just shows all of the different possibilities. Sorry, that's my dog in the background. It's going to get on his toy. Hold on. Okay. He's wondering why I took it away from him. So basically with all of these, we're just giving all of the different possibilities for what could happen in metaphase 1. So then B, it says indicate all the possible gamete genotypes that are produced in this organism as a conclusion of meiosis 2. So then it's basically meiosis 2, I believe, is the one that it has the 2 cells instead of just like the 1. And so what was the question again? Can be produced in an organism as the conclusion of meiosis 2. So at the very end of meiosis 2, you have 3 of the sister chromatids in 4 cells. And so for the first one, it's that one. The second one, this one, and this one. And then, yeah. So then C, it says how many sister chromatids are found per cell in this organism during metaphase 1. And it says, so in metaphase 1, I put that, so metaphase 1 is when all of the tetrads line up. And so I put, technically there's no sister chromatids, but technically there would be 12, but they would be connected or joined together to form 6 homologous chromosomes. So technically I wouldn't think that there's any sister chromatids at all, but if we want to get into technicalities, if they're not connected, there would be 12. And for D, it says how many chromosomes are found per cell in the organism during metaphase 2. And then metaphase 2 is when all of the chromosomes are lined up, ready to be pulled apart in anaphase 2. And so there would be 3 chromosomes all lined up and ready to go. So for 4, it says a horse has 32 pairs of chromosomes. A, it says, and then I put 64 sister chromatids just to kind of make things a little bit easier for me. It says A, how many chromosomes are present per horse cell at the conclusion of meiosis 1. And then I put 16 chromosomes are present at the end of meiosis 1 because you basically take these 32 pairs of chromosomes and you split them in half for meiosis 1. Then B, it says how many tetrads are present per horse cell during prophase 1 of meiosis. And prophase 1 is just when they're getting ready, everything gets all ready for metaphase 1 and then the rest of it. So I put, let me read the question one more time. So then the tetrads are basically the same as sister chromatids, and so there'd be 64 because that would be 32 multiplied by 2 because there's 2 sister chromatids in 1 chromosome. So for C, it says how many sister chromatids are present in a horse cell during prophase 1 of meiosis. It's basically the same question as this because, like I said, sister chromatids are the same thing as tetrads. So the 64 sister chromatids present in prophase 1. With D, it says how many sister chromatids are present per horse cell during metaphase 2 of... Did I just read the same one? No, I didn't. During metaphase 2 of meiosis. So metaphase 2 is, like we had before, is when the chromatids, they line up in the 2 different cells ready to be separated in anaphase. And so there would be 34 sister chromatids in metaphase 2. Then it says how many bivalents are present per horse cell during anaphase 2 of meiosis. So anaphase 2 is when the chromosomes, are being pulled apart. And so I said that there's none because the chromosomes are being pulled apart into tetrads. So pretty much the same as this one. So you, not really, but I mean, if you want to go technical. Then number 5 was kind of like the same as number 1, where you had to do the forked line method, which I did it, the short way this time. So I basically took, it says, consider the following tetrahybrid cross in pea plants, and then it says this one, and then it says, assume that that capital T equals tall or the dominant, lowercase t equals dwarf or recessive, capital R equals the round, which is the dominant, and the lowercase r is wrinkled, recessive, uppercase Y is yellow, dominant, and lowercase y is green, recessive, uppercase A is axil, dominant, and lowercase a is terminal, recessive. And then it says assume all of the alleles of the 4 genes are sorted independently. Anyways, so basically, to prep for this question, I did the forked line method, and so I just did the FOIL, got all of these ones, and then took the ratios of those ones into these boxes. So then for A, it says what portion of the offspring will be tall, round, green, and terminal. So then I just took the, what's it called? The letters, I'm still not sure what they're called. We're all new at this. And then I put them right here for tall, which is uppercase T, round, which is uppercase R, green, which is lowercase y, and then terminal, which is lowercase a. And then I went through down here, and then I found which ones had big T, which ones had big R, which ones had little y, and then little y, and then I put them over here and then mixed them up like we're supposed to, and then I multiplied them all out, got their percentages, and then added up there, I'm so sorry, and then I got the percentage of the offspring, which is 14.07%. So then B, it says what portion of the offspring plant has this genotype. And then again, I just went back to my already figured out ratios. Right here, I took the genotypes from there, and then calculated out the proportion, which is 3.13%. For C, it says what portion of the offspring will be heterozygous for all four genes. Heterozygous means that they're just not the same, which is these ones. And then I took those ratios, multiplied them together. That's 9, and that would be my proportion. Then D, it says what proportion of offspring will be dwarf, wrinkled, yellow, and axil. Basically, I just did the same thing for D as I did for A, except for it didn't need all of this stuff. And I just gave me a straightforward answer, which is that one. So, thanks.